cognitive function in the elderly, to subjective well-being, and in psychiatry there are well-known extreme deficits in social functioning related to autism, schizophrenia, and to a lesser extent depression. So even though it hasn't, I don't believe it's gotten enough attention within psychiatry, it's a very important trait and an attribute to try to understand. I'm in the psychiatry department, but I also consider myself a pharmacologist, and I am particularly interested in monkeys as monkeys. And social relationships and the capacity to form understanding and maintain social relationships have also been a topic of interest in non-human primates. Robin Dunbar developed a hypothesis relating the evolution of brain size and neocortical development to group size, with the argument being that it took more social capacity to keep up with the relationships in larger groups, and that this was a major driving force in the evolution of larger brains in primates and humans. That's cross-species comparisons. Individual differences within species have also been related to health, welfare, and fitness. John Capitanio at Davis, UC Davis, has an extensive series of studies where he uses a subjective questionnaire that primate observers fill out to rate animal behavior, and a factor analysis provides one factor that he calls sociability. Well, when he selects males that are high and low in sociability and uses them in experimental studies, he has a nice series of papers that demonstrate that sociability males in the home group, even after they're taken out, are more resistant to stress and more resistant to succumbing to HIV infection. And then our own Joan Silk has published a very important series of papers on demonstrating the functional significance of sociality, as Joan's term, for adult female baboons. It's long been assumed that social relationships are good for you, but there wasn't much data actually demonstrating it. But for the baboons at Amoseli, first Joan demonstrated that females who were higher in sociality had infants who were more likely to survive, and then replicated a similar effect in a different baboon population. These studies still leave a lot to be done. And what I want to talk about today focuses more on proximate causes. Why are some individuals, why are some monkeys more sociable than others? And I'm looking at life course, effects of age and sex, and for males leaving the natal group, emigration, immigration status, and for adult females, the process of having infants, all which we have some prior information or some indication that these would relate to an objective measure of sociability. For old world monkeys, the matrilineal family environment is very important for social relationships. Monkeys have preferential relationships with their matrilineal family, so it's very important to consider this in an analysis. And then my research program is particularly focused on identifying genetic influences, trait-like individual differences. And then in the last part, I'm doing something that I've just started in progress right now, and we definitely welcome any input. Stimulated by Joan's question, well, don't you have any functional data? So I'm going to try to address the question of does sociability enhance male reproductive success? Now, the approach that I'm taking is using a captive colony of vervet monkeys, the Vervet Research Colony. The animals in this colony, to the extent that we are able to, are raised in species-typical social and developmental context. We are measuring quantitative behavioral traits across the lifespan, and we're using large pedigree-based analyses to try to identify genetic influences. the colony, when it was actually out of San Fernando Valley. It was founded in 1975 by Michael McGuire, with 57 founding animals brought from St. Kitts in the West Indies. Vervet is a 3-year-old monkey found throughout sub-Saharan Africa, but there was an accidental introduction into the Caribbean during the cross-Atlantic slave trade, basically. And populations became established on St. Kitts, Barbados, and Nevis. And those populations have continued to this day, and our animals were derived from that population. We maintain a population of about 500 animals in the colony. You can see that outdoor cages. The animals are maintained in 16 stable matrilineal social groups, and females remain in the natal group with their mothers and female kin essentially for life. And males transfer at puberty, not on their own, but we take them out and then put them into new groups. We manage the population to produce about approximately 50 new infants per year by vasectomizing males to limit population growth and exchanging males at 3 to 4 year intervals between groups. And so groups become breeding groups and vasectomized groups alternately. We have descendants of 24 of the original matrilines living in the colony. In matrilines, there are 5 to 8 generations, not living animals, but 5 to 8 generations from founding animals. We have DNA from more than 1,000 individuals, and paternity determined by molecular genetic paternity exclusion for 930. And the vervets form a very complex extended pedigree. Part of this is because of the natural mating system. I should have because a male will mate with more than one female, and a male will mate with females from more than one generation. So you can see that square over there, 85, 83, is a male who has mated with 1, 2, 3, 4, 5, 6 females who are in different generations, possibly of the same matriline. You get preferential reproduction by pairing two males, and so this is a very typical picture. So it can be easy to classify relationships because it's your half-sib can be also your cousin. Fortunately, the people at Southwest Foundation have developed methods of dealing with these complex kinds of pedigrees. What I have devoted most of my time to is developing what I hope are ethologically valid behavior coding systems that reveal individual differences in traits, behavioral traits that are, on one hand, related to psychopathology, but also meaningful individual differences for monkeys within monkey society. Just collecting behavioral data doesn't mean you have a trait. You might be measuring a moment in time, you could be measuring the temporary state that an animal is in. I'm trying to find trait-like individual differences. And so I have to pay attention to, inter-rater reliability, test-retest, or time, over time reliability, construct validity. What is the construct that I'm talking about? If I use a term like anxiety or impulsivity or even sociability, does it mean what I say it means? And then in order to proceed on with any further genetic analyses, we need to determine the heritability of the traits. The measures that I'm going to talk about today are taken from the system that I call the home group scans. This is just a simple observation system that we use to track behavior of animals in their undisturbed home group. For the 16 groups, we can observe 2 groups in the morning and 2 in the afternoon. So in a week's time, we can get data on the 16 groups. And so approximately once a week, each group gets observed for, or twice a week for 24 observations in a quarter, or 72 observations in a year. During an observation, the groups are selected according to a random order list. And then the animals within the groups are selected according to a predetermined random order. And all we do is find the animal, watch them for 1 minute, and have a check sheet of behaviors, whether they occurred or did not occur during that minute, with, at the end of the minute, some information about spatial proximity to other animals. The behaviors that we code are things like, do they locomote in a minute? We measure affiliative approach, and the way we define an affiliative approach is by the simple rule, an animal goes from beyond a meter to within a meter and sits down. And this gets rid of all the drive-bys, it gets rid of the threats and or sexual interactions or any of those other things, and basically is a simple, clear-cut definition of what I call an affiliative approach. We measure groom, do the animals groom. We try to use scratching and self-directed behaviors as methods of anxiety. I'm not sure how good they are. We measure if they threatened or displayed or lip smack or submitted during the minute. We measure social play and vocalizations. In order to develop the sociability phenotype, I just selected 3 behaviors from that coding system that on their face value represent, at least to me, represent sociability. And that's just simply the sum of intervals that you either initiated an affiliative approach, you received an affiliative approach, and you were grooming, divided by the number of intervals that you're observed. And this, to me, captures the capacity to initiate, attract, and maintain social relationships, all of which are different features, but I think all of which go together into the construct of sociability. There are times of life when you're running around more, there's times of life when you're more sedentary, but are you attracting social relationships as well as are you initiating them? If you look at the scores, okay, we collected data on this system from 2001 through 2007 for, 400 plus animals per year. So if you put all that together in a mixed cross-sectional longitudinal data set, we have about 2,000 animal years. The first pass through the data is looking at each animal year as a separate piece of information. And looking at age and sex, you can see that somehow the legend got left off of this figure, but the reds are the girls and the blues are the boys. And females have higher scores on average than males. This is the mean plus or minus the standard deviation. This is a very nicely distributed measure that has broad individual differences at each time point with no 0 scores and no high outliers. It's a nicely normally distributed measure. Yeah.
Where's sexual maturation for each sex?
The males, okay, well, if you look at the blue bars, what happened at age 5 for the males? They are pulled out of the natal group, and that and they go into what we call the fraternity groups, all male groups. Some of them get selected to become new males in breeding groups. All the males at 5 and most of the males at 6 were new males thrown into a matrilineal breeding group, their first year into the breeding group. We usually put them in February, and the behavioral data are collected from April 1st through the end of December. So this is the data for the whole year. females stayed, all these females stayed in their group, and they have a very gradual decline of age. If you look at that male phenomenon and break it down into whether they're 4-year-old natal males, their year that they're a new immigrant male in the group, and then the years that they're resident males, you can see that that first year is a tough year. It's a different thing. They have to start from scratch. In almost all cases, they were put in with animals that were not from their natal group. So they're dealing with other adult males, and they're dealing with all these females. And in vervets, females join together and chase new males around. So it's a pretty trying year, but it's also a year where they're working hard to develop social relationships, which we'll show later. For females, there's suspicion from people who know monkeys, data from way back to Irv DeVore, 1965 book, a lot more recent data from Jones' work that infants are attractive and mothers of young infants are attractive. There's a lot of infant caretaking, infant handling in vervets, and so I strongly suggested that this score would be higher for females in the year that they have a young infant compared to years that they don't. And so that is a significant effect on this measure. These social context effects are not the trait-like individual differences. I think they support the validity of this measure because it's doing what it should do, what we believe it should do in these contexts. But these, if we're trying to measure a trait, we want to take out the effects of age, sex, whether you have an infant or not, and whether or not you're a male in the group. So I put the data through a stepwise regression, and I did it separately for males and females because males don't have infants and females aren't new males. So it's different, and the age curves are so different for the two. So I ran it separately for males and females, and all of those measures entered into the model. They all added, above and beyond the previous measures, and so I developed an adjusted sociability score, which is the standardized residual of the analysis for males controlling for age squared, and remembrance status in females controlling for age in age squared. Okay, so we have these measures now. Is this a trait? Well, one of the ways, because of this mixed data set, there's only a few animals that have data in all 7 years. And if I do an intraclass correlation coefficient, putting all 7 years together, I get a high value, but I lose most of the subjects. I think there's only 78 out of 704 subjects that have data for 7 years. So what I did is I broke it, I did the intraclass correlation coefficients. Now, an intraclass correlation coefficient is different from a regular correlation because it takes into account mean differences as well as relative differences. It's like an analysis of the looking at a subject effect. But it still gives you a number from -1 to +1. What I did was I put animals that had 3 years of information, 3 consecutive years, and looked at all the animals that had data from 1 to 3. And the intraclass correlation, 1, 2, and 3 was 0.6. I looked from 2 to 4, and that was 0.57. I looked because the males, very few males made that transition, the young adult to mid-adult is 6 to 8, and that was equally high. And then I did anybody who had 3 years from 8, on up, and that was high. And surprisingly enough, I didn't have a slide for it, but when you break this down into males and females, the males show just as much year-to-year consistency as the females do. So this indicates that what we're measuring here, after we take out these effects of the context, are actual trait-like individual differences. Some monkeys are more sociable than others by this measure. Now, we also have a lot of data on a different measure that that measures response to an intruder at the periphery of the home enclosure. And you put a strange monkey at the edge of the home enclosure, and we started off doing this with adult males, small groups of adult males, and we tested all the females, and then we tested all the 2-year-olds. So we have data for animals at different life stages, and you get a very strong response. You get approaching the intruder, you get sitting near the intruder, you get threatening him, you get trying to grab him, you get sticking your hand in the cage. You get a number of measures that I have used to define what I call a risk-taking or impulsivity index, because this is approaching a potentially dangerous other animal, and the animals, they show consistent individual differences in how they respond to this stranger. Well, this is a social approach that many people who do mouse models of sociability, this is how they measure sociability. So is this measuring the same thing as my measure of sociability in the home group? And the answer is no. The correlation is as close to 0 as it could be, which is good, because I never thought the Intruder Challenge Test impulsivity index was measuring something that I thought of as sociability. I thought it might overlap with the construct of extroversion, but not of affability and general sociability. So this provides discriminative validity to the measure. What we're measuring here reflects some quality of the relationships with familiar individuals. It's different from just a generalized going out into the world and approaching strangers. Now, another thing we know about monkeys, as I mentioned earlier, is that they have preferential relationships with maternal kin. In order to, in my data set, I have a lot of siblings that have the same mother. So in order to be able to look at siblings are genetically related, and they also have a shared family environment. They grow up in the same natural line. So we needed to build a maternal effect into the genetic analysis in order to be able to separate one from the other in trying to look for the genetics of this. And we use a variance components model called SOLAR, developed at Southwest Foundation by John Blangero and Laura Almasi, which basically takes the behavior matrix. We've got 704 individuals by 704 individuals. There's a correlation across their behaviors. We also know their genetic relationship to one another, and that goes in there. And then there's a matrix of maternal sharing, which is just a 0-1 matrix. So individuals that share the same mother get a 1. Individuals that don't share the same mother get a 0 in that matrix. And then there's the error variance, or sometimes called the non-shared environment. And so we put the phenotypic data into this. This is of these 704 subjects that we have. These are not 704 data points. These are 704 subjects that we have. I forgot one step. After I verified that this trait is consistent over time, then everybody gets 1 score. They get a mean score of whatever data we have from them. It's their mean of their adjusted score. So now we're down to 1 score per monkey. In this pedigree that's been going on for all these generations, we have a lot of relationships. The direct parent-offspring relationships in this sample, there's 498. You can see we have many more half-siblings than full siblings. we go down to great-great-grandparent, grand offspring. We have a few of those in the data set. And thousands and thousands of uncle, aunt, niece, nephew, the avuncular relationships, and also thousands and thousands of cousins that are either full, half, first, second, third, once or twice removed. And, so some degree of relationship. This matrix gives us a lot of power to detect if the behavioral covariance is related to the genetic matrix. And also we have that maternal component in there. It's a likelihood ratio method where we're looking at, we build different models, and it looks at the model with the genetics alone, it looks at the model with the maternal alone, and then it looks at the joint one and figures out which one is better. And for this analysis, for the 704 individuals, this model gave us a heritability of 0.53 for the sample as a whole. That is a direct translation into 53% of the variance in this measure can be attributed to genetic relationships. There was also a significant maternal effect, which accounted for 8% of the variance, which is significant, but substantially less than the genetic variance. This is important in primate studies because very often you're working with one or the other, and if you look at the maternal effect alone without the genetic effect, it accounts for more than 30% of the variance. But when you balance it out, and it's not a stepwise process. When the two of them are together, it's the independent variance that's accounted for by each one relative to the other. But this is actually fairly typical for human behavior genetic studies, that we have a sense that the family environment does more than it actually does. But so we did get a significant maternal effect. When we break the sample into males and females separately, now we lose power because we've cut the sample size in half, but it turns out that the heritability of sociability is 0.64 for females and 0.38 lower for males, but still significant for males. The maternal component is significant for females, but not for males. Well, this is the first step in a process working with Nancy Freimer, who's been developing molecular genetics resources for the Vervet, and he's developed a first-generation genome-wide linkage map. What this does is, okay, we're going to use the Vervet genomic map to find susceptibility loci on Vervet chromosomes for, whoops, that's not so, should be sociability. We're using a 2-point quantitative trait linkage analysis, again, using this program Solar to calculate it. This is a process that looks at identity by descent. So it requires pedigree relationships. It's tracing similarities in individuals who inherited the same parental allele at a given marker with their similarity. So it's using the cross-generational information. It can use population differences that might bias genetic association studies, because you can have population differences between even different areas in Europe, but particularly across broader population bases in background alleles that are characteristic of African Americans or Chinese Americans or European Americans. And so any difference between those populations could be a confound in your measure. This pedigree-based relationships does not have that problem. One of the problems that pedigree-based relationships do have is you might have restricted variability. So the pedigree might not have some types that are important. But the variation that it does have is amplified. So it doesn't give you an opportunity to find every gene, but it does increase your chances of finding genes that are active in that pedigree. We use microsatellite markers. These are high variable regions of the genome that are not functional genes, so they're not being selected against. And then we use the concept of linkage disequilibrium, where neighboring genes tend to go together to try to find regions around these non-functional markers that are related to the trait. Now, in a whole genome analysis, you're testing every location. So there's a lot of opportunity for false positives. So you have to do some really serious correction for multiple testing. So it requires very large samples. LOD scores, there's a sort of a commonly agreed on level of LOD scores above 1 are possible but marginal. Above, depending on who says it, above 2.2 is in the probable range, and above either 3.3 or 3.6 in the significant range. And this covers you for analyzing everything. In our total sample, we don't have all the markers typed for everyone because it was done in 2 different runs, and it's not easy to get things to come out exactly the same twice in molecular genetics work. So we have 260 markers. They're all being run again as we speak, so we should have 100% before too long. But so we're doing, and we're also doing preliminary 2-point linkage analysis. This is because the markers are not, they're not all in 100% guaranteed order. Many of them are, but some of them are in the best order, but not significantly determined. So we're doing 2-point linkage, and for the whole sample, for the females and for the males. And this preliminary run gave us a significant LOD score on Vervet chromosome 20, which is the equivalent of human 1, for this mean sociability score, which was replicated for the females but not for the males. It is preliminary because there's nothing around it at this point. You can see the 4 way up there on the top with just a few possibles down below. If we look at the females and the males, the females is the subgroup that's producing that high score, but we also have the highest male score, which was at a 1.5 on that same chromosome, and we have a big gap right around 100 on the base there, where we don't have any markers.
So what are we looking at on the x-axis here?
You're looking at the distance along Vervet chromosome 20 in centimorgans from the telomere. So this,
and then I get this score on the y-axis, which means what?
that's the LOD score. That's the likelihood odds ratio that this that there is a link between this measure and this region. And so one of the missing markers is in that region in there. So we'll at least know if that one does better. And then the Vervet genome is actually set up for being fully sequenced, so it'll give us information on SNPs in that region so that we can verify if this is actually a location. But it's a potentially promising location, but at this point we can't tell if it's a fluke or if it's something real. But it's still, a LOD score of 4 is extremely hard to get, and many publications in psychiatry only have 1s and 2s. So this is where we're going with that. Okay. In my last 20 minutes, I wanted to talk about what is probably of more interest to the people in this room, and that's following up on Joan's idea of looking at, does this matter for the animals? Does it affect their fitness? And and I'm focusing on males for several reasons. For one, with the females, the survival of infants is heavily influenced by management practices. But for the males, paternity is something that we really do have a pretty, it's up to them to either be the father or not be the father of the infants. So between 2001 and 2007, the 16 groups varied between 1 and 4 immigrant males per group. And then, as I said, we're trying to limit population growth, so we have between 5 and 8 groups per year that are breeding. And during this time, we produced 350 surviving infants with paternity determined. These are infants that were alive when we collect at 6 months when we first get a sample from them. There were also, there were time periods where, a 2-year period where we also got DNA from all infants that died, but that was only for that 2-year period. So I'm focusing this on the surviving infants. And I'm asking the question, does sociability influence male reproductive success? Well, across these, our sample, if we eliminate the vasectomized male groups and we eliminate any natal male paternity, which sometimes occurred in the vasectomized male groups, and focus on the groups with intact adult males, we have 45 adult male years, which are average of 3 years per male for 52 males. Of those, 4 of them, there was only 1 male in the group. But 22, there were 2 males in the group, 63, there were 3 males in the group, and 4 males. 56, there were 4 males in the group. We collect data on male rank, all occurrences of aggressive interactions between individuals. But the males, we even in the 3 and 4 male groups, we just categorize them as alpha versus other. This is the way Vervet's sorted out. I mean, there really is an alpha and other, and it's very difficult to get reliable 2, 3, 4 ranking among the other males. And there are many times when there's only 1 other male. So the alpha other both fits in with what Vervets are like and with what our data can provide. The number of surviving offspring per group year in the breeding groups varied from 1 to 16. The number of offspring per male in those groups was 0 for 57 of them, 1 for 35, and 53 males had from 2 to 12 infants. There was 1 with 12, 1 with 11, 3 with 9. I mean, it's strung out. It's not a normal distribution. And so we used this information to develop a paternity score. I call it OEE, observed minus expected over expected, where we look at the number of surviving offspring a male had divided by the number that were expected based on the number of males in the group and divided that by the expected value. And again, this is quite skewed. There are a lot of, there's a lot of low values, and then it skews out to the right for the more successful ones. Well, if we're going to look at sociability and fitness, we have to consider male rank, because rank is very important. And we believe and know from analyses that Joe has done with the Vervet data that alpha male is more likely to be the father of offspring. So the first step is to look at, well, what correlates with rank of the behavioral measures. These behaviors are all ones that are from the home group scan, including the ones that went into the sociability index, as well as some others. And it turns out, minus is just, it's hard to deal with rank, because a rank of 1 is high, and so what's really a positive correlation gives you a negative number. Alpha males are more aggressive. They're also score higher on the sociability score. And the next 3 measures are the 3 components of that sociability score. They score higher in grooming, they score higher in restraint, affiliative approach, and the initiate affiliative approach is not significant. These are all adult males. They do not differ in locomotion. Alpha males spend less time solitary, and solitary just means there's no one within a meter, and they spend less time doing the anxiety behaviors of self-directed and scratching.
Yes? Does groom include both being groomed and grooming?
Yes, yeah, and we don't split it out. In this case, I strongly suspect most of it is being
groomed.
So we've got this major confound between rank and sociability. Now, if we look at how they correlate with paternity, this observed minus expected over expected score, we see, and I threw out the 4 guys where there's only one male in the group because he's it. So now we're dealing with 141 male years, and you can see there's a very strong correlation between rank, which is coded as 1, 2, and this paternity score. We also have not quite as high, but significant the sociability score, which is the combination of the 3, and then the individual components, groom and receive affiliative approach, are all independent. If we were looking at them as separate variables, they all correlate with maternity, the male reproductive success. So how do we know what comes first and what doesn't? I put this into a stepwise regression where rank comes out first in predicting our paternity score, but groom of these individual elements added above and beyond, significantly above and beyond rank. Now in this case, by this method, rank gets credit for all of the shared variance between the 2. And it doesn't add much, but it adds significantly above and beyond what rank gets you. So, usually when I give talks, I run out of time. I go over, so I can go quickly. But this is, I am now summarizing what I've presented, that this sociability score in vervet monkeys varies by age, sex, and social context in ways that make sense. But when you control for those measures, individual differences are preserved across life stages. There are significant genetic influences on variation in this trait. There is an additional effect of maternal environment on sociability for females, and preliminary evidence for a quantitative trait locus for this trait. And then some evidence also that male sociability is related to rank and to reproductive success independently. These are the folks who collected the data, took care of the monkeys, set up the systems, did the genetic